So, someone mentioned earlier about being in the weeds, I fear sometimes I might be in the forest. So hopefully after this, I am out of the forest and maybe a little bit more into the weeds in regards to application. I don't have a whole lot of my own data to present so far, but it is my hope that after this, maybe some ideas are synthesized about how we can use what I'm referring to as the circulating phenotype. So I'm going to multiple times refer to traditional phenotypes quite a bit. That's not supposed to be a dig about anything. There are observable traits that we use every single day: birth weight, carcass traits like marbling, all of those type of traits we've used quite a bit. And so when I say traditional, it's really not trying to say anything negative about it. It's just referring to traditional versus what I'm referring to as a circulating phenotype. And I, of course, want to say that we've been able to make a lot of genetic progress. I mean, obviously, we're here at BIF, so we know about genetic progress. And we've made a lot of genetic progress with traditional phenotypes, like yearling weight on this slide, and advanced data analyses and incorporation of genomics. We've made a lot of genetic improvement. But a question, and sometimes what I think about in regards to those traditional phenotypes, is the potential limitations that we might see with those. For example, in some, there are limited biological resolution. So we don't necessarily capture the underlying molecular progress or process that's behind the phenotype that we are going for. And I am more on the molecular side of things, and I think it's important to try to merge the two. And so you'll obviously see that throughout the presentation. And oftentimes they do miss early signs of disease or stress. If we just look at our traditional phenotypes, say if an animal is sick in regards to if they come off a feed, they stop eating, their gain is different. I feel like with our traditional phenotypes, we miss some of those early signs. And oftentimes they are one-dimensional. Again, we're typically focusing on physical traits without integrating things like the immune system or metabolomics or stress responses. And so with that, I often wonder, could we be overlooking phenotypes if we're putting ourselves in a box and thinking of just traditional phenotypes that reflect both genetic potential and a real-time physiological state that both are obviously very important, but are we missing that physiological state that could be incorporated into genetic progress? And so the first part of the circulating phenotype is this is the type of components that I think of, things like steroids or cholesterol, testosterone, various proteins, glucose, all those things that are circulating with our blood. We've all gotten a blood test, you get various glucose levels, cholesterol levels, or even a complete white blood cell count. Those are all things that are circulating within our blood, and that's why I'm referring to them as the circulating phenotype. This is the first kind of level of circulating phenotype that I initially think of. You don't look at a cow and say, oh, that has XYZ white blood cells circulating through its bloodstream. You don't necessarily think that, but it is a phenotype in and of itself, these different levels. And looking at these various things, there's been a lot of important findings, and I think very interesting findings on just looking at the circulating phenotype in that sense. For example, efficient cattle labeled either low or high RFI showed lower plasma levels of specific metabolites that were used in nutrient oxidation and energy use. Infertile heifers, which is what you're seeing on this PCA plot from Phillips et al. In 2018, they actually sampled them at the time of artificial insemination, and they were able to group together heifers that did not get pregnant versus heifers that did based upon their metabolites. So that would be things such as glucose. The metabolites had a very complex name that I wasn't able to say, but they were able to predict pregnancy based upon metabolic profiles. And then also, when it comes to things like heat stress, they found that more sensitive cattle, this was done in dairy cattle, but more sensitive cattle tend to have higher sensitive to heat stress, that is, higher circulating cortisol levels before and during those heat periods. So those types of the circulating phenotype have been incredibly beneficial to being able to understand the biological processes that are ongoing within an animal that really drive some of the phenotypes that we see. So it really is a powerful tool. Blood-based tests give real-time insights. They show us what's going on in the animal right then and there, what it's experiencing, what its white blood cells levels are. They are real-time, relatively non-invasive as well. compared to other methodologies such as a biopsy of the liver, collecting a blood sample, whether you're doing that simply for a pregnancy test for PAGs, or if you're looking at metabolites, relatively non-invasive, and can be done in theory multiple times without causing the animal too much stress. And they have been really helpful for us spotting early issues as in sick or stress response. These circulating phenotypes have been incredibly useful. And I'm just referring to the things like white blood cells or testosterone or cortisol, those type of circulating phenotypes. And since we are obviously talking about genetic selection, there has been work done for heritability of those circulating traits. So they have looked at the heritability in regards to hemoglobin, monocytes, platelets, nutrients, or red blood cells number, all of those. And they estimated heritability with 2 different methods, and they found relatively mid heritability measurements for those blood characteristics. And they found strong genetic correlations between them, even if they weren't super strong phenotypic correlations, and found 91 genomic regions that were associated with the variation across those multiple traits. So they were able to find that, yes, these circulating phenotypes, such as the ones on these slides, are heritable. They are driven by genes. And so that is one of the main things that we talk about when it comes to genetic selection, is how heritable a trait is. And so they have been able to find that it is in fact heritable, these traits such as this. And so there is a lot of potential for this in regards to if we can link, okay, if they have a high white blood cell count, then perhaps they have lower average daily gain, linking these circulating phenotypes to more of our traditional type phenotypes that we think about. So I often ask myself, and maybe I shouldn't in regards to complicating things, but is there more in there? Is there more in this blood tube that we collect when we collect blood for pregnancy testing and PAGs? What else is in there other than the white blood cells and neutrophil counts? What else is in there that we could potentially use for selection and decision-based tools? Now, in order to do that, I know this probably is a review for most people, I'm going to talk about how we get from our genotype to phenotype. I do teach undergrads most of the time, so I do like to put this type of stuff in just in case, maybe you don't understand how we get from our DNA that we use for selection to our phenotype. And it is through the central dogma of biology. And first, of course, we do start with our DNA. DNA is the genetic blueprint for us, the animals. All of their cells have the same DNA in it. It is what gives the instructions. I call it a cookbook. It is the master cookbook for who we are. However, DNA itself doesn't really go anywhere. It stays in a library, it doesn't go anywhere. It's pretty protected. So we need an intermediate, and that intermediate is RNA. So RNA is the result of transcription. So imagine going into a library and check out a book, and you sit in the library and you write down what that book says. That is what transcription is. You're then able to take that piece of paper out of the library and use it how you wish. And those RNAs, there's lots of types of RNAs, which we'll see on the next slide, are incredibly important, but that is not the last step. Once the RNA has been transcribed, it goes out of the nucleus where the DNA stays and gets translated, or it might not, there's different options. It gets translated into a protein. So that's putting together blocks of amino acids that fold together that ultimately shape a protein. And then those proteins are what goes out in the cell, does its job, whether it's a cell membrane transport or it is hemoglobin. And ultimately, altogether, you get that phenotype. And so it is not just the genes there and we magically get, cows that grow faster or whatever it might be, blue eyes in this case. I know we don't care about that in cattle, but there is a process. And in my opinion, that this process offers some insights into what's going on within the body that can also be selected for. And so again, just a basic example, again, I might be preaching to the choir, but this sequence, let me see if I can, yeah, there you go. This sequence is actually the sequence for insulin in cattle, okay? So it's a whole bunch of A, C, T's, and G's all together. It is the base pairs. That does not leave the nucleus. It then gets transcribed into RNA, and then there's some things that happen, that's why it's shorter, but this is RNA. And then RNA ultimately gets translated into a protein. These are different amino acid abbreviations, and this protein gets to fold into insulin and do what insulin does in the body. So there are, typically we think about on genomics when we're talking about SNPs or anything like that, this is what we're thinking of. We're thinking of that underlying genetic sequence, but there are some intermediate steps between there that I think would be really beneficial for looking at. which is why the main focus for me is this RNA circulating phenotype. That is the RNAs that are present within our blood. Obviously, I'm not going to go over every single one of these, but there are tons of RNAs that circulate within the white blood cells in our blood, as well as just free RNA. The main one being mRNA or messenger RNA. It is that intermediate. Let me go back. It is this right here. That is our messenger RNA. It is taking the message outside of the cell to get translated into proteins. But we also have what's called non-coding RNA, which actually does not code for any proteins. It has other jobs within the cell. More specifically, ones like microRNA. MicroRNA actually has a very large role in regulating gene expression. And so there are tons of different circulating RNAs within the blood. And this is what my real focus has been on in regards to trying to synthesize some ideas in regards to how we could use this circulating phenotype in our genetic selection practices. So again, what is RNA? RNA, I say, is DNA's cousin, ribonucleic acid. It is the molecule that helps DNA turn into its proteins. Again, messenger RNA is really the one that does the main instruction carrying from inside the nucleus to outside and codes for our proteins, but there are some other types of RNA, such as microRNA, that are extremely important. And it is oftentimes, in my opinion, sometimes I think is overlooked. I love RNA versus DNA, but that's just me. I think it has a really important role. But we'll talk about why maybe it's not used as much as, say, DNA is. So this is not a novel idea. Of course, we've looked at RNA within the circulating bloodstream of many different species, as well as in cattle. Specifically, a colleague of mine at the Texas A&M Bureau, which is right next door, you'll be able to see that building this evening. They looked at circulating RNA within the blood on entry to the feedlot, and they were actually able to predict and find genes of animals that would develop bovine respiratory disease versus ones that would not develop bovine respiratory disease, just based upon their RNA levels within their blood at the time of entry. They've even used microRNAs, this one was very interesting to me, to distinguish lameness phenotypes in the feedlot. So various things like tip-toe necrosis or other types of lameness, they were able to gather a whole bunch of mRNA and it actually separated out based upon the different lameness types that they observed, as well as the likelihood of an animal recovering from a specific type of lameness. Animals that did recover and had positive recoveries had different circulating microRNA profiles than ones that did not. And one that I find super, super interesting, because pregnancy is oftentimes, we hired a new repro physiologist and he's always about how can we get our pregnancy rates up? How can we detect pregnancy rates sooner? How can we select for heifers that are going to get pregnant or more likely to get pregnant? And so a really interesting study used microRNAs, and they were able to actually at weaning differentiate between subfertile and fertile heifers, those that did get pregnant versus ones that did not get pregnant on the first round of artificial insemination. And so there is a lot of promise just circulating with cattle in regards to potentially predicting simple things, or not necessarily simple, but things like bovine respiratory disease or pregnancy or various physiological aspects like that. So the circulating phenotype has been used, again, not so much as thinking like white blood cells or cortisol levels, but it has been used and has shown a lot of potential. But where I find it very interesting as well is that RNA has a huge role in the environmental response that an animal has. So DNA is static, typically does not change within the animal, and how it's able to respond to its environment is through various changes in gene expression through RNA. So there's a whole feedback that goes on every day within an animal's life. If it experiences stressors, if it eats a different nutritional content, there are responses within the body that changes that gene expression. And then there's a feedback loop. Once the animal responds, has a certain expression level, maybe is able to produce the proteins that are needed, it then maybe turns off that gene. And so the environment plays a huge role in influencing RNA. And so that has been one of my biggest focuses, is that I think the environment plays a huge role in things. I tell my students, it's great if you have the DNA, but if you are not responding to the environment, or the environment doesn't suit, say, your DNA well enough, then you might not be able to live up to your genetic potential. And this RNA is kind of what's that mediator between that DNA and actually being able to express various traits. And so it's very, very responsive to the environment. And so, for example, altitude sickness, this was this wasn't in the United States, but it did have to do with pulmonary hypertension and brisket disease. And they were able to distinguish animals that were less susceptible. They were genetically similar in regards to and in regards to their background, but they were able to distinguish based upon RNA profiles the difference between those animals that did succumb to brisket disease and those that did not. And then, of course, temperature is a huge thing. I've already mentioned heat stress, but with the temperatures changing, they were actually able to see differences between Nelore cattle, so our Bos Indicus-based cattle, and I cannot say this breed's name, but this is a Bos Taurus cattle. These are both tropically adapted cattle, except one is Bos Taurus, and they showed distinct heat shock protein expressions both before and after a heat exposure. And so that suggests various circulating, and this is all in the blood, various circulating differences just between animals, and obviously there are different breed types, so one is better suited for the heat than the other. But those heat shock proteins are very important for heat response, and they saw a huge difference between the two, for potentially connecting things. Okay, maybe Nelore for some reason expresses their heat shock proteins more, therefore they're able to have a better heat response versus this other breed of cattle. And then interestingly enough, when they looked at RNA-seq of LPS challenged calves, they actually saw that animals that had better residual feed intake had a different response in their RNA levels than those with higher residual feed intake or less efficient animals. They actually exhibited potentially a higher immune response quicker and able to resolve it more relative than those animals that did not do that. And they were able to see that within the circulating RNA blood profiles. And most of these animals were genetically similar in that regard, other than, of course, the breed differences, but similar backgrounds, and they're able to really distinguish between what animals will respond how simply through using RNA. So hopefully by now you have some idea of why I think it's really important to focus on this circulating phenotype. There are lots of studies out there that highlight the value of mRNA and microRNA and other types of RNA within the blood and why it is something that I think should be focused on. They offer a very dynamic snapshot of what's going on, as well as being able to reflect genetic differences, which is what I'm going to talk about next. So how can we incorporate these informative RNAs into selection? Well, there's the most obvious ones that have been used quite a lot. Know or select before an event occurs. So this would be drawing blood before, and you're going to do artificial insemination, maybe before weaning, whatever it might be, and potentially are able to predict through those RNA profiles, okay, these animals need to go here, or these animals need to receive this treatment. And that's the most basic way, in my opinion, that you can use it for selection. It's real-time selection, whether that be entering the feedlot and you're able to say, okay, these animals have an elevated XYZ gene profile. They are probably going to be more likely to succumb to BRD. Let's treat them here instead of the other ones. And again, it's real-time health monitoring. Of course, we can't sample our animals, once a day when it comes to blood drawing, but it can offer a new insight into real-time blood monitoring or in health monitoring to make various selection decisions. Then I think the most basic approach if we get into genomic prediction is instead of using a quantitative trait like birth weight or weaning weight, we use RNA-seq counts as a phenotype and plug that into genetic expression programs and allow us to create EPDs for these RNA expression profiles. Of course, there's a lot of limitations to that because one, we would have to find a very, very strong signal in regards to, okay, this is a theoretical example over here. Animals with higher IL-6, which is interleukin-6 expression profiles, have better immune response, and therefore we would have to be able to make that very strong link. But in my opinion, the most basic way we can start looking at our circulating phenotype and the way that we do our, say, typical EPDs for typical phenotypes is using it as a literal quantitative phenotype in our models. And that sounds straightforward, but of course it's not. But that's probably the most basic way that we could integrate these RNA profiles, but we would have to find a very strong link between a phenotype of interest and that RNA-seq profile. And we all know who's heard of a GWAS in here. We talk about GWASs quite a bit. And so that's one of the ways we've been able to identify genetic variants that are associated with a trait. We can collect a lot of genotype data, and then we collect a lot of phenotype data, and we see, do these animals have a specific SNP or specific SNPs that influence our trait? They've been very, very powerful, but we can actually do the same thing with RNA, because RNA and its expression and its phenotype, so to say, is influenced by genetics. Variants near the promoter regions of genes, so regions of the genome that control the expression of genes, can either increase or decrease the expression of RNA. So for example, here, just a simple SNP can actually cause a transcription factor, which are kind of the machinery needed for expression, to not be able to bind anymore, and therefore that RNA is not expressed as much, or vice versa. And so any type of mutations, any type in those promoter regions, enhancers, or even within the gene can affect that RNA expression. And so there is a GWAS for expression. So instead of using a quantitative trait like birth weight for a GWAS, you're actually using the RNA-seq data as your response variable for these expression, what we call EQ2L mappings, which are expression quantitative trait loci. And so you are trying to see, can you identify regions in the genome that influence this RNA expression? And so EQ2L analysis works very similar to a GWAS. You need genotypes, you need a phenotype, which in this case is RNA. And then you run a model to find, okay, for individuals that have high levels of XYZ genes, they all have this variant. And potentially that variant is what's causing the change in gene expression between individuals who potentially have lower expression or higher expression, whatever direction it might be. And it really helps bridge those static DNA markers that we oftentimes think when we think about a GWAS with dynamic molecular phenotypes, that's circulating phenotype, and saying, okay, what does the SNP do to expression? And what potentially the next step was linking that expression to more of, say, of our traditional phenotypes. And so EQ2Ls is not a new concept. Done a lot in human research, and we've had some in beef cattle, thanks to the GTEx. Specifically in blood, a lot of the EQ2L analysis that you will look at will be in a tissue of interest, such as liver tissue, muscle tissue. But there has been some done in blood, and they were able to identify over 600,000 what we call cis EQ2Ls, which influence genes nearby, and over 9,000 trans EQ2TLs, that influence genes from far away. So 698 samples in regards to a analysis like this is not large. I think there's a lot of work to do in regards to finding more EQTLs. I need to stop saying the word. In order to be able to get a full atlas of the expression quantitative trait loci that we have. But it is a good start. And again, we'll talk about potentially why our sample sizes are so low in regards to analyses like this. And there's even more types of EQ2Ls. So the main type is looking at loci that affect mRNA expression, but then you can look at other ones too. So loci that affect microRNA expression, loci that affect methylation at particular loci within the genome. Then we have allele-specific expression, which I will talk about in my talk tomorrow if you would like to learn more about allele-specific expression. But there's more that have been relatively underutilized in regards to most of our focus has been on mRNAs and finding those that link to there. But there's a lot of other RNAs within the circulating phenotype that I think would be important to be able to map what variants are influencing what. Then another approach is what we call weighted expression networks, which essentially is almost, I wouldn't say it's exactly like a GWAS at all, but it looks at the networks that various genes have, because all genes interact, or most genes interact with each other, RNA interacts with each other. And so it looks and finds different, what they call regulatory hubs, and they say, oh, this gene is regulating a bunch of genes that are affecting weaning weight or specific trait. And so they are really good at identifying RNA biomarkers and gene biomarkers that are important in the network that is of whatever interest to the phenotype that you're looking at. And so that is, I think, a very important step that we need to take in regards to be able to use our circulating phenotype in genetic selection is continuing to link variants to gene expression patterns. So finding those genetic variants that we can identify within animals that are linked to a particular expression pattern. And so whether it be through additional EQ2L mapping or just additional mapping in regards to expression versus indels or deletions. But I think it's really important because I think we need to really prioritize relevant markers and functional markers. And that can help us refine various genetic prediction models. For example, we can use weighted genetic models that was mentioned a little bit in the previous talk. But using expression profiles and that circulating RNA to identify SNPs of interest and weighting those SNPs maybe a little bit more in our models in regards to whatever phenotype we're trying to be able to predict. We're not treating, instead of treating all markers equally, for example, we're saying we know that this SNP is linked to this gene expression, which has been seen in the XYZ phenotype quite a bit. And so incorporating that gene expression and doing either EQ2L informed weights or transcription weighted models, which we'll talk about here in a second. But we're really trying to capture that regulatory influence that SNPs have on expression and then see how that expression is affecting phenotype. So this is one of the ways we can incorporate transcription data into our genetic prediction with using these weighted models. Other approaches, so we've all maybe heard of BLUP, and then we have single step BLUP and all of the letters before BLUP. There's also what we call GT BLUP and GTC BLUP. That is genomic transcription, best linear unbiased prediction. So we actually you can combine SNP data with transcriptomic data. The transcriptomic data is an additional random effect within the model. And it accounts for both those genetic variants that we see so often used in our genomic BLUPs, as well as expression profiles. The biggest issue that they've seen with that type of model is that there tends to be a lot of overlap in between important genetic variants and important expression profiles. And so in order to try to get rid of that redundancy, there's the genomic transcriptomic covariance BLUP, and so it looks at the covariance between SNPs and transcriptome data, and that helps. We're still looking at how SNPs can regulate transcription and how they can regulate phenotypes, but it helps reduce the redundancy of the SNPs that are tied to the transcription data. These models have not been used in beef cattle very much. One of the most common papers that I've been able to find using these was actually in outbred mice. They had about a population of, I think, like almost 800 mice to use these type of data sets on, and they used the traditional genomic BLUP and then this transcriptome BLUP. And they found that the GT BLUP was actually best for phenotypic prediction and really useful when RNA reflects environmental variation. And that covariance BLUP actually had the highest accuracy they used breeding values instead of EPDs out of 9 out of the 13 traits. And in mice, when they say traits, they use things like hemoglobin, body weight, bone weight, a little bit different than what we think about in beef cattle in some ways. But those are the traits that they were looking at. And so they found a pretty good sizable increase in accuracy through just incorporating that transcriptomic data. So what is the biggest potential challenges when it comes to this type of methods? Because in my opinion, I think transcriptomics is great. I think it should be used more, but the biggest hurdle we have is the cost associated with transcriptomics. This was a recent estimate I got for a grant proposal. If you're looking anywhere from going all the way from RNA extraction from the tissue and then to your QC on the data sets and the sequencing, you're looking anywhere, depending on where you're going, to $200 to $275 per sample, okay? So it's very, very pricey in regards to what we have now in regards to genotyping. It has gone down. It's seeing the same trends in regards to decreasing. The biggest issue with RNA-seq is you do need more reads and more sequencing power to really fully capture the RNA-seq data, and that's typically what can drive up the cost. And so why we can't have, thousands upon thousands of animals with that circulating phenotype in regards to RNA, the really biggest limiting factor is cost there. However, we are combating those costs a little bit. I think we're getting closer and closer. A paper out of Troy Roan's lab did a 3' RNA-seq, which really focuses on capturing the specific part of RNA sequencing. There are shorter reads and you need less sequencing power, aka less depth. And it does tend to have lower library prep and sequencing costs and can reduce about 30 to 50% cheaper. I think they said in their paper $25 to $30 per sample, which if we could get there, that would be amazing because that's on par with a lot of our genotypic testing that we do. Another potential way to combat those prices is pooled RNA sequencing. So instead of each individual getting its own phenotype, so to say, you pool the RNA, can help reduce sequencing costs. And another way, it's not a super duper high rate of decreasing cost, but you can get genotyping data from RNA-seq data. So instead of paying for both a genotype and RNA-seq data, you can actually work backwards and get not the whole entire genome, but a good portion of the genome and then use imputation later to get a more wide coverage of the genome through RNA-seq data. So it's kind of a dual use. And so I think the most promising thing is that 3' RNA-seq. It doesn't work for all of our RNAs, so our microRNA doesn't necessarily work for, but it is promising in regards to lowering costs and hopefully getting more phenotypes in regards to the circulating phenotype. And in my opinion, if we're talking about practicality, one of the biggest issues is turnaround time. This is the case with a lot of genomic information, but it is a multi-day workflow. It can take anywhere from 5 to 10 days, depending on what lab you send it to. They could do it all in one day, but most labs are pretty swamped. So it's not necessarily real-time actionable. So when I was talking about, okay, if you're wanting to sort cattle when they come through at the feedlot based upon a certain RNA-seq, it's going to take a little bit longer than those animals are already going to go to a pen, and you're not going to want to rerun them through and sort them through again. And so there's also the issue of what we call batch-based logistics. It's a little bit more simple or sensitive to what lab is doing what and what protocol is used. And so if you have different RNA sequencing data from different places, there are ways to adjust for those batch effects, but it can get a little bit tricky. It's less uniform than our genotyping that we use. But we are, I think, finding ways to combat those turnarounds as well. Targeted RNA tests, of course, these targeted RNA tests, you need to have a target before you actually start implementing them, which typically means you have to do the more expensive things first, and then potentially come up with a targeted test that says, oh, this is going to be a SNAP test that we can use because we're targeting gene XYZ. Nanopore sequencing is also a potential. They are getting more and more into the realm of direct RNA sequencing. So sequencing RNA directly and in real time. The little MinION that they propose as a good field or shoot side can do RNA-seq. Most of the time, I don't see a whole lot of people doing RNA-seq on it, but there is the possibility for that. And then direct RNA and adaptive sampling. Adaptive sampling is something that Nanopore offers that you can actually tell the system, hey, I only want to search for these sequences. They use it a lot for microbial RNA and host depletion, but there is a potential to combine that direct RNA sequencing with adaptive sequencing in order to be able to say, hey, I want to focus on these RNAs and only sequence those, and that can help you save some sequencing power. But overall, the goal for this is really, of course, the end goal for most of us is to enhance that genetic selection, whether that be through improving accuracies or being able to identify phenotypes that we can select for in order for increased production and boost that animal performance. But I do keep in the back of my mind that we do need to keep it practical. We are working with complex biological systems, particularly when we're working with RNA. And so my ultimate goal would be to find something and be able to make it more practical for a producer to use versus a whole bunch of biological molecular processes. But there is a lot of work to be done before we can do that. But I really think we can take it a step further than DNA and approach our complex biological systems with things such as circulating phenotype and the RNA that we have. So what's next? More phenotypes. Somehow, some way, this is the biggest hurdle, and the biggest thing is because of cost. And so I think as we continue to phenotype animals in regards to this circulating RNA, we can get a whole lot more information, but that cost is one of the biggest hurdles. But I think there is a lot of promise there. And ultimately, my whole goal, I said, when people asked me what I wanted to do when I got into grad school, I said, I want to know more about how we get from our genotype to our phenotype, that lovely moniker that we hear quite a bit. And it's really finding and bridging this gap that we can get their DNA, pretty cheaply nowadays, and we can get their phenotypes, but what's going on in the middle and how can we utilize that in a better and more efficient way in order to improve our selection. And I think the circulating phenotype is a really good place to start. And with that, any questions? And yes, if you'd like full citations, please scan there. I try to do that so it's a little bit less messy, but yeah.
Thanks. Do we have any questions for Emily?
Yes.
Or I think we
Yeah.
My question. So how quickly is the expression on the circulating? Do you recommend any time point for this?
That is the biggest one. When we say more phenotypes, we're going to have to work really hard. Oh, sorry, I'll go. work really hard to, finding a time point. I think for me, one of the most interesting time points is those changes. So entering the feedlot, I'm very interested at sampling then and seeing what happens. But finding those time points, since it is influenced by the environment, it is not as predictable as, yes, our static DNA. So that's gonna be another big challenge, is when is the optimal time to be able to sample and find those time points?
Gary?
Did you look for any non-bovine RNAs within the data that you generated?
I did not. A lot of course, in humans, there's been a lot of biomarker work within circulating, but I did not look at any other, other than the bovine. And then a lot of my studies, since they do tend to have a little bit more RNA-seq data in mice, there's a little bit more out there, but my main focus here was cattle.
Back to the timing question,
Mhm.
what would you think of, could we take a blood sample on pre-weaning
Mhm.
and use that both for genotyping and circulating phenotypes?
Mhm.
And how much would that inform later in life so we could make
Yes.
decisions?
So one of my, I was fortunate enough to work with someone, they had at vaccination, pre-weaning, and then weaning again. I think the first step is looking at how things change and understanding how much things change from weaning to, because of the cost, longitudinal RNA-seq data sets are really hard to find in particularly beef cattle. So I think there's a lot of work there in order to be able to figure out, yes, we can get the genotype then, but how certain is that circulating phenotype for various traits?